Okay, let's go ahead and get started. Couple of things. The first one is a blatant plug for my lab next term, Advanced Molecular and Cell Biology Research Laboratory. If you go and look on this course schedule, that's what you're going to find. You're not going to find mutant viruses from hell. I don't think I would be able to get that through Faculty Senate. We shall see. So that's, again, next term. People can start signing up next week. Second announcement that I had, all of you know that we have a special visitor here today. She's anonymous, but I was going to force her to wear this. And if you're curious about that, I can tell you the story later. Or you can find her and ask her the story about it. So without further ado, let's start talking about mostly translation today. But before we do that, I just had a couple of quick things that I wanted to mention. You remember this ridiculously confusing slide that we had last time, looking at where the various different, small nuclear RNAs are. This is U4, U6, and U5. This is all before it comes together in the spliceosome. So you have U1 and U2, and then you have U4, U5, and U6. And then, in the interest of trying to keep this up to date, this is a paper that came out yesterday in terms of the structure now of the spliceosome once it actually binds to the intron as it involved in that reorganization step here. So I don't expect you to remember all the details here. I can't remember all of them. But importantly here is it's U6, which has all of the activity. And here's the U6 here in yellow. And the active site is right here. It's all in the RNA, which is going to be continuing through the rest of translation today as well. And then it's all of these different base pairing interactions that happen between these guys relative to what's happening here in the pre-spliceosomal state. And so that's where you need all the RNA helicase activity, all the RNA which is required for this splicing process. So yeah, we're almost up to date, 24 hours late as the case may be. So that was all I wanted to talk about in terms of messenger RNA processing. This is all about the splicing. Down at the 5' end, we put the cap on. Various cap binding complexes. Again, the proteins are going to bind to the end. It's going to help protect that messenger RNA and also allow it to get transported out into the cytoplasm. I also talked about polyadenylation. Again, this is a non-specific RNA polymerase, which just will add extra things to the end. RNA export, critical thing here is the proteins that are associated with that particular RNA after all the splicing, tailing, capping has taken place. And then we go out into the cytoplasm and we do this whole step here, translation. But before we do that, any more questions about splicing and or any of the other processing aspects? Okay, if you have more, again, week from today is what? Midterm. That means the material that we cover on Monday will be on the midterm. Wednesday will be a review. So if you have any questions that happen before then, send me an email. I'll also post them all on D2L appropriately. So in terms of translation, we are going to talk today mostly about the genetic code and eventually get down to the ribosome. One of the important concepts here are what are called open reading frames. All of us in the business just call them ORFs. And then how you go from the genetic code, which is really a nucleotide code, to the protein language. And so always people talk about translation. Well, it really is translation from a nucleotide language to an amino acid language, which are completely different on the chemical scale. And so that's that real process. How do you do the translation? You have convenient translators, who are these tRNAs. T is not for translation, though. It's for transfer RNAs. And then how the transfer RNAs get the right amino acid on them has to do with tRNA synthetases. And then the big guy, the ribosome, which does all of this job here. So let's start out talking about the genetic code. Genetic code, again, is its translation. You've got to go from nucleotides to amino acids. Amino acids, as we talked about way back when, before the first midterm, you've forgotten all about it. People thought that proteins were really the genetic material because they had so much more complexity. There are 20 of them. They had multiple different sequences. People could see that they were really different relative to each other as opposed to the really boring 4 nucleotides. Well, it turns out that the boring 4 nucleotides fit together really nicely in this kind of structure, which is probably why we're using them as the genetic information. But it brings us to a mathematical issue. And this is perfect that Ardeen is here because she's a mathematician. So she can help you with the clicker questions later. No, but what it means is if you've got 4 nucleotides, if you just had 4 nucleotides, each of those individually can't give you 20 amino acids. We just give you 4 different amino acids. So the question is, how many nucleotides do you need in a code in order to get 20? And the important thing is, of course, more than 20. So you can calculate quite easily number of possibilities. It's just 4 number of nucleotides. The number of letters in your code, 4 to the n, will give you how many possibilities. So 4 to the 1, we just talked about 4. It's not enough. 4 to the 2 is what? 16. Is that enough? No. 4 to the 4? Quickly. Way too many. 4 to the 3, however, is 64. So enough. 64 codons. So 3 nucleotides per codon. And so that's your code. You have to have words of 3 letters in order to get just 1 letter back out at the end. So that was pretty straightforward. That's basic stuff, basic math, hopefully. Is that a question or a stretch? Good. The next question, which actually was a really much more interesting one, had to do with whether the code was an overlapping code or a non-overlapping code. So what do we mean by overlapping code or non-overlapping code? That's what's shown down here at the bottom. If we start out with just this sequence, AGC, ATCG, if you had an overlapping code, you could get 5 different 3-letter words out of this. So that's a pretty efficient way to use that information that you have in the nucleotides. On the other hand, if you have a non-overlapping code, you only get 2 and a little bit back out of this. So at first, most people actually thought it would be an overlapping code. But then, through some really interesting math that we're not going to get into, it was shown that you actually couldn't use an overlapping code because it constrains all of the different kinds of combinations of the 2-letter amino acid code that you get a little bit later. So that's one reason. Another reason actually probably has to do with mutation tolerance. So we talked all about how DNA is susceptible to these chemical changes, and those chemical changes can give you genetic changes. If you have a change in an overlapping code, that single nucleotide change is going to change 3 of these codons, these words, in terms of amino acids. So a single nucleotide change can be a real problem as far as that's concerned. Whereas if you don't have an overlapping code, it's just going to change 1. And as we'll see in just a second, when we look at the genetic code, those single nucleotides often don't make a huge difference in terms of what amino acid you get, and also in terms of the kinds of amino acid at a chemical level that you end up with. So to make a rather long story short, and we'll talk more about how that happened a little bit later, people figured out what the genetic code was. And this is the genetic code. No, I do not expect you to remember it. But there are a couple of things which I will expect you to remember. And go figure, he puts red circles around them. So one of them here is AUG, which is your methionine codon. So if your messenger RNA has an adenine, uracil, guanine in it, that will give you a methionine. The other thing that I wanted to point out here is that there is a lot of variability in this whole genetic code. Some amino acids have 6 different words that will give you that individual amino acid. Most of them have 2. A few have 3. There's one other one, which is just 1. which is this tryptophan codon here. And then finally, and this was another issue that people were looking at, again in the late 50s, after they'd come up with this model for the structure, including our friend Francis Crick, they figured out that you actually also had to have ways of stopping your protein. And it turns out that there are 3 different words that say, hey, this isn't an amino acid, this is where we stop. And we'll talk more about that mechanism, how that works a little bit later on. So this is the version that we have in our textbook. I don't like this version at all. I much prefer a table version like this, because I think, oh, sorry. So, Dr. Stedman, what would you like us to know for the exam? Sorry to paraphrase your question, but yeah, the only codon that I would expect you to know for the exam is going to be AUG. That we have a variability, some have 6, and a couple have 1. If you look at some of the old exams, again, they're all on D2L. You'll see how many amino acids have 1 and only 1 codon for them. D2. So that's and then there are 3 stop codons. What they are, the individual nucleotides, I can't remember. You can look them up. I'm sure there are apps on phones and things. So I think that's not something that's appropriate to ask you to do. But again, I find that this is a much more useful way of looking at the genetic code. It doesn't list all of them out quite as easily. But this way, if you just know the first letter, for instance here, we'll have a A. And then our second letter, which is U, and give you one guess what the third one's going to be? G, exactly. So we end up with methionine. And so I think this is a lot better way to look at the genetic code for a couple of different reasons. One of the things that I really like about this is it's nicely color-coded. What does the color coding mean? The color coding means that these are amino acids with similar kinds of chemistry, similar kinds of structure. And what you'll notice is even if we do have some of these different amino acids, lysine and arginine, for instance. What's lysine and arginine? What was the important thing you wanted us to remember about that? Positively charged. You'll notice they're right next to each other in this table. And that means that single nucleotide changes aren't going to make a big difference in terms of what kinds of side chain you have. Same thing is true here for all of these dark browns. They're all hydrophobic amino acid side chains. So this nature of the genetic code, that all of the amino acids with similar kinds of chemical structures are bunched together, means that, again, just like having a non-overlapping genetic code, it's protecting against mutations that are going to then change your proteins after that. So getting back to your question you had about what's going to be on the exam. The 1 tryptophan codon, 1 methionine codon, it's AUG. There's 6 stop codons. And then there are a couple, actually it's not just leucine, that have 6 different codons here. One of the things that came up actually also quite early on, and this is another reason why people thought that the nucleotides weren't the genetic code and proteins were, is the amount of GCs varies a whole bunch, but the percentage of amino acids actually doesn't. And what that means is in some organisms, you'll use one particular codon for one amino acid a lot. In other organisms, you'll use a slightly different one. But still, the proteins are very, very similar. There's a question in the back? Sorry. Yeah, so the question is, I'm recording this, is basically, does this allow you to make minor modifications? Again, make to totally over-anthropomorphize the system, but minimal kinds of changes, but still have something which is active or just slightly different in terms of its activity, and that seems to be exactly the case. Now, how this came about in the first place, how did the genetic code evolve, is a really fascinating question. And if you're interested in it, talk to Dr. Lehman in the chemistry department, because that's exactly what he does. He also has a course, Chemistry 492, I think, where they just talk about nucleic acids. Fascinating story. Again, we could spend multiple lectures talking about it. One of the things about that, which is really fascinating, is that even though some organisms are going to use different codons for these different amino acids, almost all cellular life, with a few, very few exceptions, uses exactly the same code, which is, again, pretty amazing. And Francis Crick called it a frozen accident. Is that once you've started using this code, then you have to keep using it, because if you change it too much, then it's going to be a problem. Turns out that's not entirely true, and we found now, literally in the last couple of years, a few organisms that actually use different codons for things, and one of the most obvious ones actually here is tryptophan. So I said tryptophan has one and only one codon. That's true in most organisms, but not true in all. In some cases, actually, this stop codon ends up coding for a tryptophan as well. So we have our genetic code. How do we now turn this nucleotide business into amino acids? So the basic problem is this. Übersetzung. What the heck? So again, it's trying to get from nucleotides to amino acids. And the process of doing that was originally figured out by Nierenberg and Matai, where basically what they did is they took a synthetic piece of RNA that was just uracils, and they fed it into a little test tube that had ribosomes and everything else it needed to translate. And what did they get out? They got this whole stretch of phenylalanines. So what they did is they said, oh, well, UUU has to code for phenylalanine, right? So in that process, just continuing, making each of these synthetic RNAs and feeding the ribosomes, giving them all 20 amino acids, and then see what comes out. And that's the way the whole genetic code was figured out. So UUU is phenylalanine, and then all of the other processes that go with that. So what's figuring out that UUU should be phenylalanine? It's our little adapter ring, our translator RNA, or I always like to say, although I'm getting too old for this, what you found in the Cracker Jack box, your little decoder ring. Yes? So the question here is, if you just have UUU, how do you know then the order of the nucleotides, which is going to give you what particular amino acid? So we can get into a lot more detail about that, but it has to do with the synthesis and how you synthesize each of your RNAs. You can actually make them with different sequences, and then by back calculation, figure out what you have there. For instance, UA would be one of these RNAs, et cetera. And Gobind Khorana basically was the expert in making RNAs at the time, and he got the Nobel Prize together with Nierenberg and Matai because of this particular discovery and being able to make all of these RNAs. So yes, and if you're interested, I can send you all kinds of information about how that happened. It's also a fellow by the name of Dieter Soll, who's a good colleague of mine at Yale, who has done a lot of work in terms of figuring out the genetic code as well. So now we know, okay, that any particular stretch of nucleotides, we figured out all the words. It took about 5 years to figure out actually exactly what all of them were. And then this is great because now if you have a nucleotide sequence, you can figure out what amino acid is going to be made, or particularly one stretch of amino acids is going to be made from that particular nucleotide sequence. But there's always going to be a problem. And in fact, getting back to your question about, well, okay, how do you know what the order is of these things? And most importantly, if you're going 3 by 3, where do you start? So, we already said where we start. AUG, your methionine. And we also have stops, all these various different stops. But with any particular stretch of sequence, and this is just a single-stranded sequence. Of course, we normally have double-stranded sequences here. There are 3 different possibilities for what kinds of amino acids you could actually get out of this. Pardon me. The wrong direction here. What happens when I try and use my phone? So here, we've got 3 different possibilities. We could start at nucleotide 1, which is AUG, and gives us this stretch of amino acids. But there are also, if you start at 2, you got a bunch of stops and no methionine. So this is probably not what's going to be used. On the other hand, you could have a stretch of nucleotides that's like down here at the bottom, which doesn't have a start on it, but there might be a start over here. There might be a stop over here somewhere. So basically, it's wherever you start, AUG, and it turns out that in bacteria, it's not always AUG. You can use GUG and UUG, and even in eukarya, what happens when Stedman puts something in quotes means it's biology, so there are always exceptions. So it's almost always AUG in eukarya. Stop codons are pretty standard, again, except in these bizarre organisms that actually have some tryptophan that they use for some of these individual amino acids. And then one of the things that I like least about our textbook is that only one reading frame contains a message. That's not true. That's definitely not true, and certainly not true for the most interesting organisms ever, which are, of course, the viruses. And next term, as well as taking the mutant viruses from Hell Lab, you can take the virology lecture, where we'll talk all about these amazing organisms, etc. As far as I know, some of them actually just use 3. I don't know any of them that actually use 6, because in theory, with an opposite strand, you could actually have 3 reading frames on one side and 3 reading frames on the other, just always depending on where you're starting. So yeah, okay, we just 4 nucleotides, but depending on where you start, you can end up with a whole bunch of different potential protein sequences from all of these things. So it's not only one reading frame, and again, if I put it in quotes, that's what I mean about it. So the whole idea of a reading frame is it really depends on where you start. And if it's an open reading frame, that means you have a start codon, here again, an AUG, and a stop codon at some point. So ORFs, people are going to talk about ORFs basically constantly in my lab. We're always talking about ORFs. So open, it's just an open reading frame. Okay, any questions about this stuff? Yes? Okay, I'm not quite sure that I completely understand your question. I'll try and paraphrase it. And that is that, basically what's determining now which of those amino acids end up with this particular RNA. So it has to do with the tRNA. It's the tRNA that has nucleotides at one end, which we'll talk a bunch more about after the clicker question. And then the amino acid that gets attached to it. And we'll talk about that just a second. Yeah? Mm-hmm. Okay, so that's this is a great question. And basically, again, I'll paraphrase it. In your open reading frame, if you have 2 AUGs, which are in the same frame, i.e. each set of 3 nucleotides, can you have a methionine in the middle of your protein? And the answer is most definitely yes. And the reasoning behind that we'll talk about on Monday, because it has to do with initiation. And it turns out initiation is actually slightly different in terms of the tRNAs than an internal one. Yeah? That is exactly correct. So any of these open reading frames have a start, and they don't have a stop. And you could even say that, again, down the bottom here, this is clearly an open reading frame if there was a start over here somewhere. Here, these aren't, because they don't have any of these potential starts, although there might be one over here off on the other side. Yeah? Yeah, so these are, sorry if I didn't explain that properly. So the question is, basically, where do you start? Where do you start reading on any of these things? It's a reading frame, but where do you start with it? This thing. Keep clicking through here. So basically, it's just which nucleotide you start as your first one. And so here, you started with nucleotide 1. Here, you start with nucleotide 2. Here, you start with nucleotide 3. And how that happens, again, we'll talk about on Monday. Turns out to be very different between eukaryotes and bacteria, which is really interesting. Okay, so now finally, we get our clicker question? Yes? So, if there are only 2 nucleotides per codon, how many different amino acids could be encoded in a non-overlapping genetic code? And Karen, you're not allowed to answer this one. Could have lent you a clicker, though. I'm trying. And again, I'm sure there are one or two of you who are just trying to screw up my statistics. I have all the scores up here, by the way. I can see who's, 10, 5. Say, I think you people are messing with me. So yes, it's, what? 4 to the n. 4 squared is 16. Yes, thank you. I'm gonna run you people down who came up with these other ones. So. Yeah. So we should ask our dean if that's a good idea. Okay, so let's talk a little bit about these adapter rings. The way that you can go from your nucleotide to your amino acid. It's all about the translator RNAs or the transfer RNAs. This is what people almost always think about when they think about a transfer RNA from 5' to 3'. A couple of things about tRNAs. A, it's really nice to draw them in these structures, but that's of course not what they really look like. That's just a two-dimensional description of them. We'll see the 3D structure on the next slide. But a couple of other things which are really quite important, and that is that these tRNAs, they're made by what polymerase in eukaryotic cells? They're almost all made by RNA polymerase 3. What does RNA polymerase 1 do? Makes a ribosome mostly, which we'll talk about again right at the end today. So in these RNAs, which are made by, again, RNA polymerase 3 for the most part, they're transcribed, but then there's a bunch of changes that happen to them. So these are all post-transcriptional modifications that happen. So transcription is going from your DNA to RNA, but then a whole bunch of modifications that happen. And in the case of this alanine tRNA, it's going to have an alanine attached up at this end, but also lots of modified nucleotides. So for instance here, I don't expect you to remember all these names, dihydrouridine, methylguanine, dimethylguanine, this funky psi over here, which stands for pseudouridine that we talked about last time in terms of modifications in ribosomal RNA. And then we'll talk a lot more about inosine down here at the bottom. This here, from 5' going to the 3', this is your what's called the anticodon loop, and that's because these are the 3 nucleotides which are going to base pair with the codon and give you that translation saying, hey, this is the interaction here. Here are the 3 nucleotides, and then our amino acid, which is up here at the top. Yeah. Okay, so the question is the anticodon of the 3' end of the structure. The 3' end of the structure is where the amino acid is added. The anticodon is almost in the middle of your sequence. Varies a little bit between different tRNAs, but it's more or less in the middle of that. So the anticodon we'll talk about. So the question is, what's the function for the anticodon? It's binding and base pairing to the codon, but we'll look at that more in just a second here. But before we do that, I did want to show what the structure really looks like, and that's this one right here. So whenever you show these sort of nice base pairs one across from each other, of course they're not like that. They're in nice double helical form, not unlike your DNA. And you also have a number of base pairing interactions that happen between these various different helices. So here, our dark blue is up at the top, the gray, they're opposite each other in the cloverleaf diagram, but they're still clearly interacting with each other. And our anticodon is down here. Here's where that amino acid is attached up here. So again, what's the anticodon doing? The anticodon is interacting with the codon through base pairing interactions, and it is true with all of the base pairs we've talked about in this class, and certainly true in our DNA, is they're always anti-parallel to each other. So 5' to 3' on one strand, 3' to 5' on the other strand. So that's the interaction that's taking place. So that's one thing that happens. But one of the other kind of surprising things that people found is how many potential codons do you have in your genetic code that are not stop? 64 minus the 3 stop codons. And yeah, no, Karen, you can't answer that one either. So we have 61 possible tRNAs, right? That would be matching these anticodons. Turns out they're actually way fewer than that. In some cases as few as 30, often about 50. So how the heck can you have these 61 possible codons? You just skip some of them? You don't even use them? Well, actually, that's not the case. And this is something that Francis Crick also came up with. He said that, well, maybe there are some tRNAs that don't have to perfectly base pair with each other. Maybe we can have one of those nucleotides that's a little floppy or wobbly. And so basically what he said is that the first position of the anticodon, so anticodons with your tRNA, doesn't necessarily have to have a perfect match with the third position in your codon. And if you remember the genetic code, this thing I didn't emphasize, but very often you'll have a block in that table format where all of them are exactly the same amino acid. And what that means is that the third nucleotide is not important. We're going to end up with the same amino acid, doesn't really matter. And so that's what Crick came up with, and it turns out he was exactly right in this case. You can have normal UA base pairing, you can have UG base pairing, and in many cases you have this strange modified nucleotide. What's I? I is inosine. So it's a deaminated adenine that we'll look at a little bit more depth later. But it turns out that this is particularly good at making base pairs to 3 different codon sequences. So inosine at the first position in your anticodon can base pair with 3 different nucleotides at the third position. Yeah? So I'm not completely sure I understand your question. Maybe let's go back and look at this structure. This is maybe a better way of looking at it. So the anticodon's down here. Yeah, so that was the theory at the time. So, okay, let's just repeat the question. Does this allow you the flexibility to have wobble interactions, which are going on there? And so Francis Crick proposed this in the early 1960s, that this is what was happening. When people finally got high-resolution structures of the ribosome associated with the tRNAs, which was about 10 years ago, so about 40 years after this, they found that was exactly the case, that there was, in fact, a little bit more flexibility at that first position of the anticodon relative to the messenger RNA when it's actually in the structure of the ribosome. Okay, so take-home message here. Same tRNA can recognize more than one codon. So that's how you can get away with less than 61 different tRNAs. tRNA modification, as I mentioned before, one of those is inosine that we'll talk about in just a second here. But many, many, many different modifications are happening to your tRNAs. One thing that happens is tRNAs get spliced. Now, this is not a splicing reaction like what happens with the spliceosome. These are very specific endonuclease ligases that will cut out individual pieces of your tRNA And interestingly enough, it turns out that almost all of these introns in tRNAs are at or near the anticodon loop, so the purple that we have here. So those are getting spliced out, and even in some organisms, what people found is that sometimes these tRNAs are actually encoded in two different halves and then get brought together inside the cell in terms of making these tRNAs. So splicing is happening. Another thing that happens is your primary transcript, just like we had with our messenger RNAs and the ribosomal RNAs, they are longer than the final tRNA. They get chopped at the end. This chopping process actually happens also through an RNA. And in some cases, you're going to add a couple of nucleotides at the end here where that amino acid is going to get attached as well. Other tRNA modifications that take place seem to be important for quality control in terms of making sure that the tRNA is associating properly with not only the codon, but the amino acyl tRNA synthetase that we'll talk about in just a second. And if you're interested in more details about this, talk to Dr. Water-Royal in the chemistry department. We've got a very strong group of people working on RNAs that are here. Some of the modified, yeah, sure. Yeah, so the question here, again, sorry to paraphrase, is that one of the reasons that you're not having the same kind of spliceosomal activity with these tRNAs that you would with the messenger RNAs that you get from RNA polymerase II is that, yeah, RNA polymerase III doesn't have all of that spliceosomal machinery with it. So these are separate enzymes that are doing this. And these are all protein enzymes, and that's what the colors are down here. It's actually that protein enzyme that's doing this. So it's not all about the RNA here. It's much more about the individual splicing reactions. So what are these modified nucleotides? My favorite one is actually down here. It's not in the textbook. It's called archaicine. It's got this massive amine group hanging off at the end here. Again, this is something that Dr. Water-Royal works with in terms of what's going on in his lab. We've had some research grants to work on this. But the other ones we've talked about so far are dimethyl G. We get a couple of methyl groups added here. Dihydrouridine, which is where you've added across this double bond. Thiouridine that we didn't talk about. And then probably the most important of these is inosine. So inosine, instead of having your normal NH2 down here, you just have one H, which is at the bottom. So these modified nucleotides and the modification process that has to happen turns out to be really important. Some of these modified nucleotides are actually brought in with your diet, and some of them are not being made properly in some people with genetic diseases. And so these modified nucleotides are extremely important. But I'd like to concentrate on inosine. Again, this is our normal adenosine, which is down here, would have an amine group, which is hanging off here. Inosine is missing that amine. Sorry, it should be right here. And it can base pair with cytidine, uridine, or adenosine. So here is our tRNA at that first position, again, always anti-parallel. You have inosine in many cases, and then two other nucleotides. These nucleotides are almost always going to base pair in a normal way, whereas in the wobble position, sometimes you'll have I, and in a couple of cases, you can actually have uridine, which can base pair with G. Eukaryotes seem to be a little bit more stringent in terms of what they use, whereas bacteria is a bit more wobbly and loosey-goosey, as it were. So that's how we're getting interactions. So getting back to your question about codon-anticodon interactions, it's through this pretty normal base pairing with, again, the exception of that wobble base. So that's great. That's what's happening at one end of your tRNA. But what's going on at the other end of the tRNA? Yeah, so another way of putting this is very, very well said, is another way of thinking about this is inosine is masquerading as some of these other bases. because it can then sit there. But it has to be at that wobble position. Because if you try and put inosine at the second or third position of the anticodon, it can't do that base pairing anymore. Okay, so how does the cell know that with this base pair interaction, you have a particular amino acid? Sorry.
On this slide, it's saying deaminated adenosine.
Mm-hmm.
On the previous one, it said deaminated G.
Yes.
Is it two ways to get to the same thing?
Okay, so the question here has to do with how do you get to inosine? And this actually has to do with the biosynthesis of the whole process. So if you just think about inosine and what it looks like in terms of the chemical structure, it's a deaminated adenosine. But biochemically, how it gets made, it actually comes from guanine. So that's where the confusion lies. But as far as we're concerned, I wouldn't bother too much about that. Okay, so finally, how do you put the amino acid on? Well, how you put the amino acid on is through these wonderful enzymes called the aminoacyl tRNA synthetases. Really a rather large mouthful, but basically what these proteins do is they figure out basically what both ends of your tRNA are doing. They bind down here at the anticodon loop, and then they put the appropriate amino acid onto the 3' end. So this is a very specific interaction between this tRNA, the aminoacyl tRNA synthetase, and putting on this particular amino acid. In theory, you could have just as many aminoacyl tRNA synthetases as you would have amino acids, but also in terms of the different tRNAs. Turns out that in most cases you only have 20 aminoacyl tRNA synthetases. In some cases actually even less than 20 aminoacyl tRNA synthetases. And what that means is, since the lowest number of tRNA genes that are out there is about 30, that some of these aminoacyl tRNA synthetases actually can interact with multiple different tRNAs. The important thing is really what amino acid gets put on the end here. So you can have different anticodons, but still always have exactly the same amino acid, because many of these aminoacyl tRNA synthetases are what are called isoaccepting. They can bind to multiple different tRNAs. Now, they're very similar tRNAs. They can have very similar sequences here because the genetic code is really similar. And then the amino acid, which gets added here, is the very last thing. I have quotes on 20. Why quotes on 20? Whenever 20 means it's not quite exactly right. There are a couple of cases. One very well known in bacteria, where it turns out that there's an aminoacyl tRNA synthetase that puts on the wrong amino acid, and that then gets changed by a second enzyme afterwards. And it turns out that this is even more extreme in the archaea. Some of the archaea only have 17 aminoacyl tRNA synthetases, and then 3 extra enzymes that are going to add and modify that nucleotide at the end. Again, why this is? Do we ever ask why questions in biology? No. It's there, and quite why it is, we really can hand wave and speculate here. A great person to ask these why questions to is John Perona, who's a chemistry professor also here. And in fact, I think this particular structure here was in fact done in his lab before he moved to PSU. So what is aminoacyl tRNA synthetases doing? Well, they're putting an aminoacyl group onto your tRNA. But there are 2 steps whereby this happens. And this is very similar to what we talked about again way back at the beginning. You need to get activated precursors in order to do assembly of macromolecules. So it's not just putting on the appropriate amino acid onto the particular tRNA, it's also making an activated precursor that can be used to make proteins later on. So the first thing that these aminoacyl tRNA synthetases do is activate an amino acid. So they take an amino acid, here's our favorite amino acid with a R side chain, again, could be any of the 20 here. Through hydrolysis of ATP, there's a AMP that gets added to your amino acid. So adenylated, this is your amino adenylation. And then this adenylated amino acid gets added to the 3'OH that's at the very end of your tRNA. And now this is a active ester bond. This is a quite unstable bond, and so this is providing the energy for translation, which is going to be happening a little bit later on. So two activities of the amino-acyl tRNA synthetase. One is activating the amino acid. The second is adding it to the appropriate tRNA. So appropriate tRNA here is tryptophan, for instance, gets added to through this high energy bonds. There's a slightly different way of looking at it. And now we're going to have the interaction again. So the 3' end, 5 to 3 base pairing. And now we're ready to have the translation take place because we have all of these, what are also called charged tRNAs. A charged tRNA has this activated amino acid attached to the 3' end. There are lots of problems, but one of the most major problems here is that amino acids, and I'm sure all of you remember your amino acid structures, some of the amino acids actually look really, really similar to each other, from a chemical point of view, extremely similar to each other. And amino-acyl tRNA synthetases make mistakes. They'll put on the wrong amino acid occasionally. And so how does that get fixed? Turns out that there's more proofreading. So we already talked about proofreading in terms of your DNA polymerases, minimal amounts of proofreading in terms of RNA polymerases. Now we've got proofreading in terms of translation, but this is happening at the level of the amino-acyl tRNA synthetase. So we saw the structure a little bit earlier on. High resolution structure had the amino acid basically right here. Amino acid gets added based on this structure of your tRNA. What happens here is this gets added, and sometimes it'll actually be the wrong amino acid that gets added, but it's only going to be a slightly wrong amino acid. Isoleucine versus leucine, for instance. It's just got a methyl group in a slightly different place. Phenylalanine and tyrosine. Phenylalanine doesn't have an OH on it. Tyrosine does. So these are the common kinds of mistakes that take place. So there are two different enzymatic sites here. This is above and beyond the amino acylation step, where this potentially wrong amino acid can get cut off. And so it's a lot like what happens in terms of the DNA polymerase. If there's a slight problem, that then can get chopped off. And it turns out the specificity here is actually specificity for the incorrect amino acid. So we'll take, for instance, the phenylalanyl amino-acyl tRNA synthetase. If it puts on a tyrosine, that tyrosine can go into this editing site and get cut off. Phenylalanine, however, cannot get put into here. And so we can look at some of these examples right here. Already talked about isoleucine and valine. It's all about this extra methyl group here. Phenylalanine, all about an OH, which is here. And it's that error correcting or proofreading mechanism here that gives you about a 1 in 1,000 error rate. Well, 1 in 1,000 is not great. What is DNA polymerase error rate? With all the extra stuff attached to it, mismatch repair, et cetera. About 1 in a billion, actually, with mismatch repair and proofreading, et cetera. So translation is not great in terms of this specificity. But translation, if you do make it wrong once, probably not going to be as critical. And that particular amino acid you add is going to be pretty similar in terms of its structure. So it's unlikely to really mess with your protein too much. And there are quality control that happens. We'll talk about those later in terms of getting rid of the proteins that are not actually properly functioning. So this is the proofreading that happens in the amino-acyl tRNA synthetase. But what about the ribosome? Is there another step of proofreading? Yeah. Okay, so basically, sort of restating things, you have, in most cases, you're going to have 20 amino acyl tRNA synthetases, which you're going to put on, and they're different, so each of those is going to put on one different amino acid. And those that could be putting on very similar amino acids, like here, isoleucine, valine, phenylalanine, and tyrosine, these are ones that have editing sites that will take out the incorrect one. Yeah?
So, in bacteria, when they have when they have a codon for a different amino acid, is it in the amino acyl tRNA synthetase where the difference is taking place?
Yeah, I'm not sure I completely follow your question here, but since the genetic code is almost completely universal, these are going to be the same kinds of interactions, and the amino acyl tRNA syntheses are extremely similar in all kinds of organisms. I'm not sure if that completely answers your question, but... Okay, so that's okay. The question here is basically how universal is genetic code is another way of putting it, and the answer is it's almost completely universal. There are few variations in very few bizarre bacteria and mitochondria, which actually are slightly different, and they do have different tRNAs which are being used in those cases. But it's only, again, these are really rare cases. Okay, so what about proofreading by the ribosome? So when stuff actually starts to get put together. And the answer is the ribosome does absolutely no proofreading. The only quality control has to do with the tRNA and the amino acyl tRNA synthetase. So all the proofreading is happening there, and it turns out this was proved really quite nicely by taking a cysteine tRNA and just changing that cysteine to an alanine, and it turns out that the ribosome puts in the wrong amino acid. So it'll have a cysteine codon there. If you've changed all the cysteines to alanines on the tRNAs, you have a whole bunch of alanines instead of a whole bunch of cysteines. So there's zero proofreading that happens in terms of the ribosome. It's all about the amino acyl tRNA synthetases. More questions? When you ask more questions, what does he mean? Take out your clickers, please. So I'm not sure that the dean will be able to help us too much with this one. So the post-transcriptionally modified nucleotide inosine is present in some tRNAs at which of the following positions? The 3' end of the codon, the 5' end of the codon, the 3' end of the anticodon, the 5' end of the anticodon, or A and C. See if you can get 100% on this one. Ten. 5. Lift off. Okay, we seem to be split about half and half. The 3' end of the anticodon or the 5' end of the anticodon. So, how do these nucleic acids interact with each other? Always anti-parallel to each other, right? So, the 5' end of your codon is going to be what? The starting one. That's going to base pair with what? The 3' end of your anticodon. So the wobble position, which is the third position of your codon, is going to be what? The 5' end of the anticodon. So I warned you when we talked about 5' and 3', it's going to rear its ugly head many, many, many times. Okay, so now we've got our amino acyl tRNA. It's ready to go. Now how the heck do you actually make protein? Well, the way that the protein gets made is, again, through this activated precursor that we talked about, that first activity of the amino acyl tRNA synthetase. When it's added that to the end of your tRNA. So here's our amino acyl tRNA right here. What this does, together with the ribosome, is now we have the N-terminus of this individual amino acid, which is now going to attack the C-terminus of the amino acid, which is bound to the other tRNA here, and in that process is going to make a peptide bond right here. But this whole peptide is still attached to this new amino acyl tRNA. So what happens is your protein gets built on these individual tRNAs. And this is what the ribosome does, catalyzes this reaction where you end up extending through here. So the process of making your protein, N-terminus is the beginning, the C-terminus is the end. Again, a lot like all of our polymerases, the nucleotide polymerases, which do what? What direction do they go? 5' to 3'. So N-terminus to C-terminus is analogous to your 5' to 3'. And it turns out it's exactly the same way that it's going along the messenger RNA as well. So it's reading in the same direction. And these are, you can also talk about this being a ribonucleoprotein because we've got our ribonucleotide here and the protein which is hanging off the back of it. So what is this? Translation. So we've got all of our pieces. How do they all come together? Just like you have with replication, just like we have with transcription, we have 3 stages: initiation, elongation, and termination. Today, we're just mostly going to talk about this elongation step. We'll talk about initiation and termination on Monday. Another way of looking at this cycle is here, just doesn't have all those nice labels on it. So initiation, the really important thing here is getting your tRNA, and it turns out this is a specific tRNA, getting back to your question about methionines in the middle or methionines at the end. So it's a very specific tRNA that binds to that first AUG that you have where you're going to start translating from. That interacts with the small ribosomal subunit. The small ribosomal subunit is interacting with the messenger RNA and basically bringing everything to the right place to get your initiation. Once you have the small subunit and your initiator tRNA, then you have the large part of the ribosome that comes on. Then you have extension. We just talked about that reaction here, transpeptidation that happens, building your peptide chain, hanging on to this amino acid here till you get to the end, and we'll talk about that again, and this whole process goes around and around. So what do I mean by large and small subunits of the ribosome? You've probably seen all of these S's before. Hopefully you've seen them all in principles. 30S, 50S, 70S, etc. How does 30 plus 50 equal 70? Yeah, biologists are really bad at math. No. So it's a good thing that we have our dean here. Help us out with this. No, but this has to do with the sedimentation coefficient. So the S actually stands for Svedberg, who's a Swedish scientist who came up with the ultracentrifuge. And basically, this defines the both shape and size of anything which is there. So if you purify ribosomes just by themselves, it turns out they're going to sediment at a particular position. For prokaryotic ribosomes, bacteria and archaea are relatively small. The eukaryotic ribosome is a little bit bigger, and so it's going to sediment a little bit further. But you can separate these really quite easily into the large and small subunits, and that turns out what you need to do. So your initiation is always getting that small subunit to your messenger RNA, and then the large subunit is going to associate with that. Small subunit is going to be your decoding part. That's the part that's initiating with your messenger RNA. The large subunit has the activity, which is the transpeptidation activity. These are made up of multiple different RNAs, all modified through all the SNO RNAs that we talked about last time. Different sizes in terms of the bacterial ribosome. We've got 2 different RNAs which come together here, 16S and 23S, a bunch of proteins in either case. We'll see a little bit later that most of those proteins have nothing to do with the activity. They're all about holding everything together, so it should sound really familiar based on what did we start today's lecture with? The spliceosome, also proteins, are just holding things in the right place. And so all the activity here, it's all about the RNA. And any of you who have taken microbiology have probably heard a lot about 16S RNA or the small subunit RNA. When we looked at the very beginning of the class about the 3 different domains of cellular life, bacteria, archaea, and eukarya, that's based on the sequences of these small subunits of the ribosome. Because this is found in all cellular organisms. All of them need these particular ribosomes. So what do they look like? This is it's actually the very first, in fact, crystal structure of the ribosome, showing here the gray, which is all of your RNA, and then these yellow things, which are all the proteins that are hanging out on the outside. This structure was published in 2000, so 17 years ago now, but at that time was by far and away the largest structure of any biological macromolecule which had been found. This is and almost no one remembers, it's actually the archaeal ribosome subunit. I have to remind people of that. In eukaryotic cells, there are about 1 million of these ribosomes. So if you remember, if you go back and just look at cells in terms of how much RNA is present in those cells, the vast majority of that RNA is the ribosomal RNA. And that's because you've got a bunch of these ribosomes which are actually present inside the cell because you have to have it for translation here. A lot of people talk about the ribosome as a ribozyme. We talked about ribozymes last time. What's a ribozyme? So enzyme which is made of RNA. So we're not going to have a chance to get to it today. Right at the very end, I have a link to a video about Harry Noller. He was, actually still is, a professor at UC Santa Cruz who basically showed that it's just the RNA. The RNA and the RNA alone is enough activity to give you transpeptidation reactions. And because of that, he got a little prize at the end of last year called the Breakthrough Prize. Only $3 million for studying this, thanks to the Google people and everyone else. But this is just the RNA in the large subunit of the ribosome. And it turns out that all of these different pieces, you look at these now, and their secondary structures are all extremely well conserved. You find them in all kinds of different life. And one of the things that, as I mentioned before, Niles Lehmann is doing is looking at these ribozyme activities in terms of how the very first life could have been happening. How could you have a molecular system which has enzymatic activity, RNA has enzymatic activity, and have some genetic activity that goes with it. So a lot of people talk about the ribosome as being sort of a molecular fossil because it's what's left from that time when we just had RNA as a functioning part of our system. So we'll talk more about the ribosome next time. Have a good weekend.